You're cleaning out the attic on a weekend, or maybe just walking past a blooming tree, and you breathe in this huge lungful of dust or pollen.
Which happens to all of us.
Right, exactly. And for some people, their lungs just quietly clear it out and they go about their day. But for others, that single breath triggers a full-scale, suffocating allergic attack. I mean, the chest tightens, the wheezing starts, and the airways just completely locked down.
Yeah, it remains one of the most frustrating biological overreactions we deal with because, the trigger itself, like pollen or pet dander, it's largely harmless, yet the body's response is just catastrophic.
And for a long time, the microscopic chain of events that bridges that gap, moving from a harmless particle to a catastrophic airway shutdown, it's been missing a few crucial puzzle pieces.
It really has. But we finally have some answers.
We do. Today on The Deep Dive, we're looking at a groundbreaking 2026 paper published in the journal Cell that fills in those pieces. Our mission today is to decode a completely new, honestly shocking mechanism behind allergic airway diseases like asthma.
And the main culprit driving this biological explosion is actually a surprisingly heavy element, iron.
Iron.
Yes.
Which is crazy. So we're going to treat the epithelial cells lining your lungs like a microscopic crime scene today. We are tracking the exact sequence of events from the millisecond an allergen enters your airway all the way to a full-blown asthma attack.
So to understand this crime scene, I think we should first look at the weapon the allergen is actually using.
Okay, let's unpack this. What are we looking at?
Well, when we talk about allergens, whether that's papain from papayas or proteins from house dust mites or even proteases from aspergillus fungi, they don't just passively bump into your lung cells. They actually possess enzymatic activity. They're basically biological scissors that actively cleave structures on the cell surface.
Wait, so the allergen isn't just acting like a chemical key fitting into a lock to trigger a response?
Not at all. It's much more destructive.
Yeah.
The lung cells are coated in a receptor called PAR1, which stands for protease-activated receptor 1.
Okay, PAR1.
Right. And the inhaled allergen acts directly on this receptor, snipping it at a very precise location, specifically the amino acid residue R46.
It's literally severing a trickwire.
Functionally, yes. That is exactly what it's doing. And because it's a physical cut at R46, it's completely irreversible.
Wow.
So once that PAR1 tripwire is severed, it sets off this massive intracellular alarm, which initiates a highly specialized process called ferritinophagy.
Ferritinophagy. Okay, I want you listening to visualize this for a second so we can remember these terms.
Yeah.
If we imagine the lung cell as a factory, you have ferritin, right?
Correct.
And ferritin acts like this heavy-duty steel vault made up of FTH1 and FTL chains. Its whole job is to safely lock up dangerous reactive iron.
Yeah, that's a great way to picture it.
And then you have NCOA4, which is sort of like an armored transport truck. So when that PAR1 alarm gets tripped, the NCOA4 truck grabs the ferritin vault and drags it straight to the cell's incinerator, which is the autolysosome.
I love that analogy, especially the incinerator aspect, because once the autolysosome melts down that ferritin vault, all the iron trapped inside is suddenly liberated.
Just dumped into the cell.
Exactly. The result is a massive flood of labile iron, which just means free, unbound iron. And the researchers found this happening incredibly fast.
How fast are we talking?
Within just 30 minutes of the initial allergen exposure.
30 minutes. But how do you actually prove that chain of events in a lab? Because tracking free iron flooding a single microscopic cell sounds incredibly difficult.
It is, but they used a very structured, graded evidence approach. First, they measured the iron levels directly using ICP-MS, which is mass spectrometry.
Right.
And they combined that with a fluorescent dye called ferro-orange, which basically lights up when it binds specifically to labile iron.
So they can literally see the iron levels spiking massively.
Right. But to prove causality, to prove that this specific tripwire pathway was responsible, they had to throw some roadblocks into the factory.
To see what stops the flood.
Exactly. They started with a drug called Vorapaxar, which inhibits that PA1 receptor.
So the scissors can't cut the tripwire.
Right. When they added Vorapaxar, the allergen couldn't sever the R46 residue. The tripwire stayed intact, and the iron flood never happened.
Wow.
Then they moved downstream. They used inhibitors like chloroquine and baphyllomycin A1 or BAFA1, which completely shut down the autolysosome.
So in the factory analogy, they turned off the incinerator.
Yeah, exactly. So even if the tripwire was cut and the truck transported the vault, it couldn't be destroyed. The iron flood was completely prevented.
Okay, let's pause and lock this in for you because that was a lot of science. Here's what we have so far in this timeline. Number one, inhaled allergens act as scissors, and snip the PAR1 tripwire on the surface of your lung cell.
Yep.
Number 2, this alarm tells the NCOA4 armored truck to drag the ferritin iron vault into the cellular incinerator. Number 3, the vault is melted down, instantly flooding the cell with free iron. It really makes you wonder, have you ever thought of your respiratory allergies as a literal rust problem, like an iron management issue deep inside your lung cells?
It's a huge shift in perspective,
Right.
especially when you consider how incredibly dangerous that free iron is.
Right.
Labile iron is highly reactive. Left to its own devices, it would cause widespread oxidative damage to all the cell's internal structures.
Which raises a big question. How does the cell survive this flood long enough to actually trigger an asthma attack? I mean, it seems like the cell should just self-destruct immediately.
You'd think so, but the cell survives because the iron is immediately grabbed by a specialized escort. It's an iron chaperone protein called PCBP2.
PCBP2.
Yeah. Its entire job is to capture that dangerous labile iron.
Mm.
And the researchers used Gaussian accelerated molecular dynamics, or GSDMD simulations, to map exactly how PCBP2 behaves.
And the structural shift in that simulation is just fascinating.
It really is.
Because when PCBP2 is empty, it maintains this very relaxed, wide, flat shape. But the moment it binds the free iron, the simulation shows the protein physically contorting. It snaps into a tight, tense, path-focused shape.
Yeah, it's quite dramatic.
It's like a baseball catcher's mitt snapping tightly around a fast-moving ball.
That is exactly what it looks like. And that conformational change, particularly in a region called the KH3 domain, is what dictates the protein's next move.
Because it's not just holding the iron, right?
Right, it's transporting it. Now that the catcher's mitt has the iron secured, it navigates to a very specific target protein called gasdermin D, or GSDMD for short.
Okay, and this is the part of the paper that seems to break a major biological rule. Because GSDMD is a well-known protein in immunology, right?
It is, yeah. But the way it's being handled here goes entirely against conventional understanding.
How so?
Well, ordinarily, GSDMD is activated when it gets cleaved by enzymes, specifically proteases from the caspase family. Biological scissors, basically.
Right, the usual way proteins get cut.
But the researchers tested an array of protease inhibitors to see if they could stop the asthma pathway here. They used caspase inhibitors like E64 and ZVAD, which absolutely should have stopped GSDMD from being activated.
But they didn't.
In this allergy model, they did absolutely nothing. GSDMD was still being cleaved, regardless of the enzyme inhibitors.
Here is where it gets really interesting. How do you slice a protein in half without the biological scissors, the proteases?
Yeah.
If enzymes aren't doing it, what is physically cutting GSDMD?
It comes down to raw, explosive, localized chemistry.
Wow.
Remember that PCBP2 chaperone? It docks the iron directly onto very specific amino acids on the GSDMD protein, residues E309 and Q312.
Okay, so the iron is parked right on the protein.
Exactly. And concurrently, because the lung cell is under stress from the allergen, it begins naturally producing hydrogen peroxide, H2O2.
Oh, wow. So you have iron and hydrogen peroxide mixing right on the surface of the protein.
Yes. And that initiates what is known as a Fenton reaction. But because the iron is physically tethered to GSDMD, this isn't some random chaotic reaction spreading through the whole cell.
Right.
It's a highly localized Fenton reaction. It generates these incredibly constrained hydroxyl radicals.
Functionally, the cell is building a microscopic chemical bomb right on the target.
That's a very accurate way to describe it. Hydroxyl radicals are incredibly volatile. And because they are generated directly on the surface of GSDMD, they immediately attack the nearest delicate structure.
Which is the protein itself.
Right. They chemically blast apart the peptide bonds at a specific sequence of amino acids from 309 to 313. The ELRQQ sequence.
So the chemical blast breaks the protein into its active fragments without needing an enzyme at all. That is just a brilliant piece of cellular engineering.
It really is.
But wait, how did they confirm that it was this specific explosive chemistry causing the cleavage and not just some undetected enzyme they missed?
They proved it by systematically removing the components of the reaction.
Ah, okay.
When they introduced NAC, which is a ROS scavenger that essentially mops up explosive free radicals, the cleavage stopped.
Makes sense.
Then they turned to genetic mutation. They mutated the PCBP2 chaperone, specifically creating a D82A, E168A, and E355A mutant, so it can no longer bind the iron at all.
And what happened?
The cleavage stopped, then they mutated GSDMD itself, the E309A and Q312A mutant, so the iron couldn't dock onto it. And again, the protein remained entirely intact.
So the cleavage absolutely requires that localized iron peroxide reaction.
100%.
Okay, time for another quick recap to keep this complex pathway clear for you. So, bullet 1, the PCBP2 catcher's mitt grabs the free iron. Bullet 2, it docks that iron onto the GSDMD protein.
Yep.
Bullet 3, the iron mixes with hydrogen peroxide to create a localized Fenton reaction, a literal chemical microexplosion. And bullet 4, this explosion blasts GSDMD into its active form without any enzymes at all.
Perfectly summarized.
Does it surprise you, the listener, that your body uses explosive chemistry, literally creating destructive free radicals on purpose, as a precision signaling tool?
It really highlights how tightly regulated these cellular mechanisms usually are, but also, how catastrophic it is when they are weaponized against the host during an allergic response.
Yeah.
Because the fallout from this microexplosion is what ultimately drives the physical disease.
So what does this all mean? What actually happens to the cell after GSDMD gets blasted apart? Where do those fragments go?
Well, the critical piece is the N-terminal P40 fragment. Once it breaks off from the blast, it is drawn straight to the cell's outer membrane.
Okay.
Multiple P40 fragments aggregate there, and they embed themselves into the lipid bilayer, linking up to form a physical pore straight through the cell wall.
Basically punching microscopic holes in a water balloon.
Exactly. And at that point, the cell starts leaking its internal contents out into the surrounding lung tissue.
I'm guessing what leaks out isn't good.
No, it's not. The most consequential molecule that leaks out is interleukin-33. Or IL-33.
IL-33.
Yeah. In immunology, IL-33 functions as an alarmin cytokine. It operates essentially as a biological distress flare.
Okay.
So as IL-33 washes out of those P40 pores, it hits the surrounding immune cells, specifically activating group 2 innate lymphoid cells, or ILC2s.
The ILC2s are basically the immune troops responding to the flare.
Exactly. And the response is overwhelming. The ILC2s begin rapidly pumping out downstream signals, primarily IL-5 and IL-13.
Ah, I recognize those.
Right, because this is the exact transition point where microscopic cellular chemistry manifests as the physical symptoms of asthma.
So this is where we feel it.
Yes. IL-5 and IL-13 drive eosinophils to infiltrate the lung tissue. This triggers severe airway inflammation while simultaneously forcing the airways to overproduce heavy, thick mucus.
Wow. That's the chest tightness, the wheezing, the physical inability to get a clear breath. It all traces back to those tiny pores punched in the cell membrane. But the paper doesn't just stop at mapping this pathway, right? The researchers set up an in vivo mouse model to see if they could actually intercept this process.
They did. They wanted to establish an algorithm of care, basically testing whether intercepting the iron could prevent the entire downstream cascade in a living organism.
So what did they use?
They utilized iron chelators. These are drugs specifically designed to bind and remove free iron.
Okay.
Specifically, they use deferriprone, which is DFP, and cycloperox, CPX. These are compounds typically utilized for severe systemic iron overload diseases.
So they're testing these heavy-duty iron sponges as a potential asthma intervention.
Exactly. They administered DFP directly into the airways of the mice prior to the allergen exposure.
Yeah.
The results were definitive. The gelator basically sequestered the entire labile iron pool. So without free iron, the PCBP2 mitt had nothing to transport.
Wow.
GSDMD never underwent the Fenton reaction, the P40 pores never formed, and the IL-33 distress flare was never released.
So the asthma attack was neutralized before it could even begin.
Completely. No eosinophil infiltration, no airway inflammation, no excess mucus.
That is incredible.
And to really solidify the mechanism, they also ran the experiment in reverse.
What do you mean?
They administered iron dextrin, or ferrous sulfate, to a separate group of mice, artificially increasing the iron load in their lungs before exposing them to the allergen.
Oh, I see. So they supplied more ammunition for the Fenton reaction bomb.
Exactly. And the resulting allergic inflammation was exponentially worse. The cellular damage, the mucus production, the immune cell infiltration, everything was massively amplified compared to a standard allergic response.
Wow.
It confirmed that the sheer volume of available iron dictates the severity of the inflammatory cascade.
Okay, let's bring it all home with our final breakdown. Bullet 1, the blasted GSDMD fragments punch holes in the cell membrane. Bullet 2, the IL-33 distress flare leaks out and activates the ILC2 immune troops. Bullet 3, this causes the actual mucus and inflammation of asthma. And bullet 4, mopping up the lung iron with chelators like DFP, completely stops the whole disease in its tracks.
Nailed it.
So if you or a loved one suffer from asthma, how does it feel knowing the secret to calming those airways might not just be about blocking histamines or, using broad steroids, but about soaking up microscopic iron?
It really demands a reevaluation of our therapeutic targets. Asthma management has historically focused on mitigating downstream effects, like using bronchodilators to open the airways or corticosteroids to suppress the broader immune response.
Right, treating the symptoms.
Exactly. But this research identifies a highly specific upstream bottleneck. If we can regulate the cellular iron economy within the epithelium itself, we might prevent the inflammatory cascade from initiating in the first place.
It is just remarkable to consider the sheer density of the biological machinery operating in the background. Every time you step outside on a high pollen day and take a breath, there's this entire microscopic relay race executing in the delicate lining of your lungs.
It's a lot going on.
Seriously, allergens are cleaving PRR1 tripwires. NCO4 armored trucks are hauling ferritin vaults to the autolysosom incinerator. PCB-P2 catcher's mitts are snapping around free iron, delivering it to GSDMD to detonate a localized Fenton microexplosion.
It's quite a visual.
And all of this is unfolding in fractions of a second, entirely invisible to us, dictating whether the airway remains clear or just clamps down.
It is a stunning display of molecular precision, but, it also introduces some concerning variables when you consider the environment we are currently living in.
And that is a crucial, provocative point to leave you with today. We've traced how intracellular iron acts as the necessary spark for this inflammatory explosion, right? And how artificially increasing that iron load severely exacerbates the asthma pathway in the mice. But if we pull back from the microscopic view and look at our modern environment, our cities are completely saturated with airborne particulate matter. Environmental pollutants like smog, industrial exhaust, and especially brake dust from traffic, they are heavily laden with transition metals, including massive quantities of iron.
Yeah, that's the unmentioned implication here. If the underlying mechanism of severe allergic airway disease is fueled by iron availability, we have to evaluate those external inputs.
Okay.
Is the modern skyrocketing rate of urban asthma simply a biological reaction to natural allergens, or is it being fundamentally amplified because we are literally breathing in the exact metallic catalyst required to drive this explosive cellular chain reaction?
It is definitely something to think about the next time you're stuck in heavy traffic with the windows down. Thank you for joining us on this deep dive into the hidden architecture of the lungs. Keep questioning the world around you, keep looking closer, and we'll catch you next time.